So in section 4.2, we are going to describe the structure of DNA. We are going to explain the Sanger sequencing method, and we are going to talk about the differences, of course, between eukaryotic and prokaryotic DNA. Let's start with the structure of DNA. Okay, 3 components. You have nitrogen, and that is a nitrogen base. You have carbon, there will be 5. And it is a sugar, a pentose, meaning 5, a 5-ring pentose, and a phosphate group. Now, one of the things that you will need to be able to do is count the carbons. Little fun from organic chemistry. This is carbon number 1. 1, 2, 3, 4, 5, 6. The most important ones are this 3' carbon and this 5' carbon, because this will be how they are connected. This is not as inherent when you look at the ring structures. It is not as easy to count the carbons because you don't have the structure that looks like this. But just know, this is how we're counting the carbons. 1, 2, 3, the 3' carbon, 4, 5'. Okay? That will be important as we move along. The nucleotide is named based on the nitrogen base. The nitrogenous base can be a purine, which are these, such as adenine and guanine, or a pyrimidine, such as these, which are cytosine and thymine. We're not going to talk about uracil quite yet because that is RNA. And always, just like Chargaff's rule from the last chapter, you will have a adenine paired with a thymine and a guanine paired with a cytosine, a purine paired with a pyrimidine, but very specifically A and T, G and C. Here is what that looks like in different ways of organic chemistry, if this helps you in some way to think of it that way. Now, how did we get here? Again, a series of experiments. We already know from Chargaff that A goes with T, so we knew a purine paired with a pyrimidine. There were several things that we did not know. So, controversially, Rosalind Franklin worked in what she believed to be was her own lab, what Dr. Wilkins believed was his lab, and it did not go well. Honestly, they didn't like one another. You can watch Photo 51 for extra credit, and it will explain this entire situation. But Rosalind Franklin took this picture, which is known as Photo 51. Watson and Crick, unbeknownst to her, stole that data from her. As she was leaving the lab, they took her data and were able to puzzle how the DNA was done in a helix. You can sort of see this helical structure here, and this is the X-ray diffraction picture taken by Rosalind Franklin. So Watson and Crick actually found this picture because it was stolen by Wilkins and given to them, and then they were able to determine the structure of DNA. Her Photo 51 article was published at the same time as a reference, but not as a, hey, we stole this data. And it has since been discussed, and now you will often see Rosalind Franklin mentioned more predominantly. So Watson and Crick proposed that DNA is made up of 2 strands of phosphorus, essentially, that are twisted around to form a right-handed helix. Again, mostly from Photo 51. The pairs take place between a purine and a pyrimidine, which they knew by Chargaff's rule. So they figured out this structure, and then they figured out that there was a phosphate backbone. And that is how they came up with the structure of DNA that you see here. There's also a major groove, the bigger one, and a minor groove. Until the 1990s, the sequencing of DNA, reading the sequence of DNA, was a relatively expensive and long process. Just for some context, my entire PhD thesis took me 3.5 years, looking at these things individually, reading them off, A, T, C, G, And now my entire dissertation can be done in about 2 days. Good times. So essentially, that process that I did manually can now be done using radiolabeled nucleotides, which made it much easier and much safer. So essentially, here's an electro gel, and here is someone loading the gel, which again was done by hand up until recently. With currently available technology and automated machines, the process is cheaper and safer and can be completed in a matter of hours. Fred Sanger actually developed the sequencing method, which was used for the Human Genome Sequencing Project, which part of it was done at WashU here in St. Louis. Let me show you this video. Okay, here is the video.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain.
Do you see how that works? Isn't that cool?
This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
Okay, so do you see how that goes from the A to the T? Do you see how the Sanger sequencing method works? And then you will get something that looks like this based on the fluorescent and the deoxy chain termination. You will get something that looks like this. Again, something that looks a little bit like this is how it is often thought of, and it first has to be denatured, right? It has to be split apart so you can get to just one sequence. Then that DNA, right? First you have to denature it, unzip it. Then you have to make a whole bunch of copies, like the video we just saw. Then you have to attach a primer. We will do this in class, but you have to attach something to the sequence that you already know is there. If there's a bunch of AAAATs, the primer will go there. So it doesn't zip back up, and so it can start sequencing. You will add a polymerase solution to it. You will grow a complementary chain into looking at the termination dye. You will denature these growth chains, and then you will use electrophoresis to figure out what the chain is. The tubes are labeled ATGC, and according to the nucleotide used, that is what you will use for the each of the 4 di-deoxynucleotides, and it will carry a different fluorescent label. This chain elongation continues until a fluorescent di-deoxynucleotide is incorporated, after which no further elongation takes place. So you'll get all of these fragments. Then every difference in length until you get the entire thing, right? So the first one is a T, and then there's something in another T, and then something in a C, and then that's how you get the sequence. You will see these large fragments, and it will look like this on a sequencing machine. For his work on the DNA sequence, Sanger received the Nobel Prize in Chemistry in 1980. This is an overview of what that process looks like. And just a refresher on what gel electrophoresis is. It's a technique used to separate DNA fragments of different sizes. So the gel is made of agarose, just like an agar plate. Once the gel has solidified, the DNA is loaded on the gel and an electrical current is applied. The DNA has a net negative charge. DNA has a net negative charge. So it moves from a negative electrode towards the positive electrode, and it moves by size. So it is applied and sufficient time to let the DNA separate according to the size. The smallest fragments will be the farthest from the well. They will move the furthest because they are small. And the heavier molecules will be closer to the original well, right? The smaller it is, the further and the faster it can go. And the larger it is, the slower it goes and the closer it stays to home, if you will. Once the DNA is separated, the gel is stained with the DNA-specific dye and it is viewed. That is the Sanger sequencing method. Now, let's talk real briefly about prokaryotic and eukaryotic. We already know that the prokaryotic nucleoid is just folded chromosomes. There's not the nucleus and all of the chromatin and the histones. That is not all happening in the prokaryotic cell. So most prokaryotes contain a single circular chromosome that is found in the cytoplasm, and it's called the nucleoid region. There's a bigger picture of that. In prokaryotes, the DNA is sort of twisted in what's known as supercoiling, so it just sort of twists around, and it's sort of underwound or overwound. If you think about a slinky and how you would wind that up, some proteins are known to be involved in the supercoiling. Other proteins and enzymes such as DNA gyrase, which we will hear in both DNA in prokaryotes and eukaryotes, the gyrase helps maintain the supercoiled structure. It helps it sort of tie up and make it smaller. So again, the entire DNA has to be twisted in order to sort of fit it into the area that it needs to go, even in prokaryotes. Now, this is even more true in eukaryotes. So in eukaryotes, whose chromosome each consists of linear DNA, employ a slightly different strategy. Instead of just winding it around itself, it's packed around what are called histones. At the most basic level, DNA is wrapped around proteins known as histones, and that's how it compacts itself into these chromosomes. So the DNA, remember, it is negatively charged because of the phosphate group, is wrapped tightly around this histone core. This is called a nucleosome. The nucleosome is linked to the next one with linker DNA. Now, this is very confusing because this is still DNA. It may or may not code for things. And when we start talking about the histones and uncoiling, we will talk about this linker DNA and how tightly this is wound around here, and maybe things that are exposed or not exposed based on how tight this is wound around that histone and what is allowed to be transcribed and translated. But just for now, we need to remember the DNA is wound around histones, which make a nucleosome, and in between that is something called linker DNA, which moves it together, and this is how it is tightly wound into what is known as chromosomes. These are also called beads on a string, a beads on a string structure. With the help of the fifth histone, a string of nucleosomes is further compacted into a 30 nanometer fiber, which is the diameter of the structure. Metaphase chromosomes. So what we're talking about here is the way the chromosomes look when they are in metaphase, what we kind of think of as what a chromosome looks like. And that's the metaphase chromosome. They are even further condensed by scaffolding proteins to be what we would historically think of as a chromosome. And at that metaphase stage, the chromosomes are at their most compact, approximately 700 nanometers in width. So just an overview of what this looks like. You have DNA. That DNA is wrapped around histones. Those are called nucleosomes. Those nucleosomes are coiled into chromatin fiber, and those chromatin fibers are further condensed until you get it so condensed that that is what you look at in the metaphase chromosome. Okay, so let's review that. The currently accepted model of the double helix of DNA was proposed by Watson and Crick with help from Rosalind Franklin's data. Some of the features are there are two strands. It's made up of a helix, and it has a complementary base sequence that is anti-parallel, meaning an A always goes with a T, a C goes with a G, and a pyrimidine is always with a purine. There's alternating deoxyribose sugars and phosphates form the backbone, and the bases, A, T, C, and G, are the rungs of the ladder, paired with one another. Again, a purine always pairs with a pyrimidine. A pairs with T, G pairs with C. One turn of the helix has 10 base pairs. Prokaryotes are much simpler than eukaryotes. Most prokaryotes contain a singular circular chromosome, and in general, eukaryotic chromosomes contain a linear DNA molecule packed into nucleosomes like this, linear, wrapped around histones, formed into chromatin fibers, ultimately making this condensed chromosome. So, overall, can you describe the structure of DNA? The three parts? Can you explain the Sanger sequencing method? And can you discuss the similarities and differences between how the prokaryotic DNA is just supercoiled and the eukaryotic DNA is wound around histones and then into chromatin?